Hi, my name is Kim Wever. Welcome to my web lecture about internal validity. Have you ever wondered how to assess the quality of a research paper? Would you like to know more about internal validity and risks of bias, especially in preclinical animal studies? Then stay tuned, because that's the topic of this web lecture. After attending this lecture, you should be able to achieve the following learning outcomes. Please take a moment to read them, and let's revisit them at the end and see how you did. Before we set off, let's get our definitions straight. We're going to talk about study quality. But which aspect of quality do we actually mean? A lot of different terms come to mind, such as power, precision, and reproducibility. But in this talk, we'll focus on one of them, internal validity. Internal validity is a key aspect of quality for experimental studies in which a group of subjects undergoing an intervention, such as a drug treatment, is compared to a control group. When assessing internal validity, we ask, are the results of this study valid for the population in this study? If a study is internally valid, its results reflect the cause-effect relationship between the intervention and the outcome that's measured. Now, this may not always be the case, because internal validity may be threatened by bias. Bias is a systematic error. It's caused by a variable other than the intervention of interest, which influences the outcome and causes differences between the experimental group. So, bias makes the data of one group slide or shift systematically into a certain direction, which distorts the experiment's outcome. Let's have a look at different types of bias that may affect the study. Different types of bias can occur in different phases of an experiment. To have a closer look at the different types of bias that we can encounter over the course of a study, let's visualize our experiment as a balance beam. We imagine that we jump onto the beam here at the start of the study, and our goal is to balance all the way to the end, where we reach unbiased results. But along the way, we come across several experimental phases where bias may occur, causing us to fall off the beam and reduce the validity of our study's results. The sources or causes of bias are different for each phase of the study. We differentiate between selection bias, performance bias, and detection bias. Let's examine them one by one. Selection bias occurs before doing any experimental procedures, all the way at the beginning of the experiment, where you select which subjects are included in the experiment and in which experimental group they will be. Selection bias occurs when an error in the selection process causes systematic differences between the groups in the baseline characteristics of the subjects. When these baseline differences affect the study's outcome, they systematically distort the results. Let me show you some examples. Imagine an experiment where the population of interest is fruits, and you're picking subjects from a variety of options. In an unbiased experiment, subjects in the two groups should have comparable baseline characteristics. We're happy. But if you don't take measures against selection bias, you may unintentionally end up with unequal baseline characteristics. For instance here, all the red fruits accidentally ended up in one group, while the other group contains no red fruits. The color of the fruits may affect your outcome measure, leading to distortion in the study results. When your selection criteria are more narrow, for instance if your population of interest is apples, there likely is less variability in baseline characteristics, but differences between groups can still occur if no measures to avoid bias are taken. Let's take a moment to address the difference between variability and bias, because these are sometimes confused. Some methodological aspects of a study can cause variability between subjects within the groups. This can happen, for instance, when selecting subjects of a wide range in age or weight, or when a researcher is inexperienced in performing a certain procedure. High variability reduces the precision of the study's results. However, as long as factors causing variability affect all groups in the experiment to a similar extent, they do not cause bias. Only those aspects of methodology that lead to systematic differences between the groups cause bias. Now, let's look at a real-life example and think of measures to reduce bias in selection procedures. In the following clip, you will see a selection process where mice are assigned to two experimental groups. What strikes you when you observe this procedure? Do you have suggestions for improvement? Okay, so I hope you noticed two things about this. First of all, picking mice from a box like this may seem random at first glance. But if you look carefully, the experimenter actually picks up the mice in order of their tendency to run away. The slow, perhaps less anxious mice are picked up first, while the jumpy, fast mice are picked up last. Since the first 5 mice are all allocated to the intervention group, this causes systematic differences in baseline characteristics between the groups, which poses a serious risk of selection bias. To avoid this, the animals should be assigned to the experimental groups in a truly random order. For instance, by using a computer-generated random number sequence, by rolling a dice, or drawing lots using blinded sealed envelopes. Secondly, the experimenter here has knowledge of which mouse gets assigned to which experimental group, which may cause him to unintentionally influence his decisions. For instance, if he expects the intervention to be stressful for the mouse, he may prefer the more docile animals to go into this group. Blinding of the experimenter to the group labels would further reduce the risk of selection bias. Now that we have selection bias covered, let's move on to performance bias. Performance bias can occur throughout the performance of the study, during day-to-day care of the animals, as well as during experimental procedures. It occurs when there is a factor other than the intervention of interest, which differs systematically between the experimental groups and influences the outcome. Let me illustrate it a bit further. Back to our imaginary experiment with 2 groups of 3 apples. In one group, we apply the intervention of interest, which is exposing the apples to a pair of teeth. The other group undergoes the control intervention using a toothless dragon. When we go on to measure how many bites have been taken out of the apples, we want the difference between the groups to be caused only by the intervention of interest, not by any other factor in the experiment. The true effect of the intervention may be distorted if some factor, for instance the performance of the experimenter or an environmental factor, systematically differs between the groups and influences the outcome. Let's look at some examples and think of measures to reduce this type of bias. Let's do a quick assignment. Suppose you walk into an animal room in your facility and notice cages of animals arranged on the shelves like this. Why is this probably not the best way to house the animals? Do you have suggestions for improvement? The answer lies in the environmental factors in the animal room. Apart from the intervention of interest, these groups now also differ according to the position of their cages in the room. For instance, there are differences in light intensity and temperature between the top and the bottom shelf, which can influence animal behavior and drug metabolism. Other factors include visibility of the animals to the caregivers, this is better on the shelves at eye height, and proximity to the door, which means more or less disturbance from people entering the room. How would you reduce the risk of these factors introducing performance bias? I hope you agree that randomizing the cages across the shelves, or having animals from different treatment groups in each cage, would be important measures to reduce the risk of performance bias. But wait, there's more. Here's another example of a situation in which performance bias can occur. What's the potential problem here? And what measure would you suggest to avoid performance bias? The potential problem here is that the researcher or caregiver is not blinded to the experimental group this animal is assigned to. Recall that in the previous example too, the cages were labeled with the group assignment. Obviously, this researcher is acting out of concern for the well-being of this animal. But if his actions differ systematically between the experimental groups, this can introduce bias. So blinding and randomization are both essential to prevent performance bias. I hope you're ready for the last phase of the experiment, the outcome detection. Detection bias can occur when measuring outcomes, either during or at the end of the experiment. Again, when there are systematic differences between the groups in the way outcomes are detected, this can lead to bias and distort the study results. Let's have another look at our apple experiment. Of course, after we induce an intervention effect, our aim is to measure this effect as accurately as possible. Similar to performance bias, if some other aspect of the detection method differs between the experimental groups, the outcome we measure can be systematically distorted. The detection bias can lead to an overestimation or an underestimation of the actual difference between the groups. Here are some examples to think about. This pathologist is assessing histological slides. Can you spot the two issues with this methodology? What would be your solution? And how about this second example? I hope you noticed that in both examples, the pathologist is assessing the slides one group after another, and as a result, his fatigue, or his learning curve, can cause detection bias. Secondly, he's not blinded to the group allocation of the slides, which can influence his assessment based on his expectations of the treatment effect. Again, randomizing and blinding the outcome assessment is essential to prevent bias. Well done! Now let's see how we're doing on our balance beam. We've come a long way from the start of our experiment. Now we have only two more types of bias to avoid. The first one is attrition bias, which can occur throughout the study. Let's have a closer look. Attrition is a gradual reduction in number, and attrition bias occurs when the number or the reason of dropouts, aka subjects lost to follow-up or incomplete outcome data, differ between the experimental groups. Dropouts can occur throughout the study, for instance during selection, where some animals may not enter the study to begin with, after disease induction, when some animals may die, at outcome detection, when samples are accidentally lost, or during statistical analysis, when outliers are removed. In all cases, attrition bias can distort the study results. Here, if all green apples are lost to follow-up in one group, but not in the other, this causes an artificial systematic difference between the groups in the color of the apples. Note that attrition bias is often aggravated when new subjects are added to replace the dropouts until a certain number of subjects is reached. But why is attrition bias so important? Here's a video scribe from the folks at Sketchy EBM explaining this further.
Hey, welcome to Sketchy EBM. I'm Anthony Crocco, and today we're talking about Lost. Sadly, we're not talking about a mysterious South Pacific island with all our friends Hurley and Sawyer and Jack and all the rest. Today we're talking about Lost in the context of research. Specifically, we're going to talk about loss to follow-up, and this is something that shows up in randomized controlled trials, especially trials about prognosis. Because I think it's important for us to really understand the impact of patients being lost to follow-up, I want to go through a simple example. Now, this is obviously a fictitious study with a ridiculously low number of participants. The overall N of this study is 4, with 2 participants in the control group and 2 participants in the experimental group. In the control group, one of the 2 participants has the good outcome, and the other participant does not have the good outcome. Now, I'm a bit of a math whiz, and I calculate the probability of good outcome in the control group to be 50%. In the experimental group, one participant has the good outcome. Unfortunately, the second participant was not able to be reached by the researchers, and as such, they weren't able to know really what that person's outcome was. So the only results that the researchers have for the experimental group is the one participant who did well, and gives us a rate of 100% for this group, a number to me which seems not entirely appropriate. Let's imagine what might have happened to this participant and how that might affect the study's results. First off, the missing participant might have had the good outcome, which would have given the experimental group 2 out of 2 with a good outcome, or 100%, making it better than the control group. Secondly, the participant may not have had the good outcome, and this would have left the experimental group with a 50% probability of good outcome, in which case it's no better than the control group, and we'd have to weigh the other things involved with making a decision, such as cost, compliance, and adverse effects. Lastly, on top of not having the good outcome, the participant may have had some horrible adverse effect. This would make the control intervention the better choice. I hope this illustrates to you why participants who go missing in a study really mess with our ability to make any conclusions about that study's results.
I hope you see now why attrition matters so much. Sometimes dropouts cannot be avoided. But to minimize the risk of attrition bias, it is essential that the same criteria for inclusion and exclusion of subjects, samples, or data points apply to all experimental groups. And if dropouts occur, the reason and the number should be reported per group so that the risk of bias can be assessed. We've got one last type of bias to cover in this talk. Let's go back to our balance beam. Fantastic! After successfully avoiding selection, performance, detection, and attrition bias, we have achieved unbiased results. Of course, we want to share this with the world. But since researchers communicate through publications, we need to take one extra step to ensure internal validity: unbiased reporting. So the final type of bias I'd like to tell you about is the bias that arises from selective outcome reporting. Bias due to selective outcome reporting occurs when not all outcomes that were measured in a study make it into the publication, and the decision whether to report them is based on the nature or the direction of the results. Not reporting data for certain outcome measures, time points of measurement, experimental groups or subsets, or certain statistical analyses are all examples of selective outcome reporting. For example, outcomes for which no significant effect is found, or those that contradict current theory, are often not reported. Selective outcome reporting is suspected to be one of the most substantial biases affecting internal validity. As a side note, selective outcome reporting is a type of reporting bias. There are many others. The most well-known is probably publication bias, but most types of reporting bias happen between publications. Selective outcome reporting occurs within a study, which is why it's relevant for internal validity. Let's have a look at a real-life example. Let's say that a certain researcher performed an experiment, gathered the data shown here, and submitted the manuscript to a journal for review. One of the reviewers suggested only reporting some of these data in the publication. Which data do you think the reviewer suggested to leave out? So here's what happened. The reviewer suggested to only show the data which showed significant differences between the groups. This would mean removing the 24 hours time point entirely, because no signal was detected yet, and all intervention groups which showed no effective treatment. But doing so would have biased the report by omitting valid and relevant data. Without them, we get a biased impression of the effects observed in the study. Plus, publishing them can be very important to prevent unnecessary duplication of experiments. Luckily, the reviewer could be convinced to change their mind, and the entire dataset was published. So remember, just because data are neutral doesn't mean they aren't useful. An important measure we can take to prevent and detect selective outcome reporting is pre-registration of study protocols. Registering a protocol before the start of a study helps with thorough preparation, recording for instance details on blinding and randomization, but also which outcomes you intend to measure. As readers and reviewers, we can compare publications to the initial protocols to find out if any outcomes are changed or missing. Preclinicaltrials.eu is an international register of preclinical trial protocols you can use. And a number of journals now offer registered reports, publishing study protocols in advance, as well as the resulting manuscripts after study completion. So that's all, folks. We've reached the end of our balancing act. Wow, I feel good. I knew that I wouldn't. Thanks for your attention. I really hope you enjoyed this lecture. Here are some messages for you to take home. Now that you know all about internal validity and risks of bias, maybe you'd like to learn how to assess risks of bias in a publication using a designated assessment tool. If you do, then please join me for another lecture soon. See you later!